Tantiana Bronich. I consider her to be my mentor for the past 4 years. She is currently the professor of Department of Pharmaceutical Science at our institutes. She's also the co-director of drug development and nanomedicine here. Her talk today is nanotechnology for radiation oncology.
Thank you. So good afternoon, and of course I would like to thank the organizers for their kind invitation. But most of all, I would like to thank organizers and all speakers for absolutely great opportunity which the symposium present for myself, for my education in this area. And I would like to share a couple of points in a little bit area which is a little bit far from radiation oncology per se, or clinical, maybe immediate clinical application. And we would like to talk about the area which is growing quite rapidly in the last 20, 30 years. And it's related to the how we can apply nanotechnology materials which are of nano size or nanostructured, how they can be applied to the medicine. And there is a different definition of the nanomedicine, but I think that the most important one, could we find the unique properties of such materials which can be applicable to the medicine? I would like to say that this field of the research is extremely interdisciplinary. Right? Nanomedicine doesn't exist itself, so it's actually symbiosis of many, many efforts from the different areas of the science and engineering, and starting from the chemistry and physics and going through the extremely deep understanding of the biology of disease. And then when it's applied to the certain aspects of the manufacturing and fabrication technique, only in this case we can come to whatever we name nanomedicine. So, and I would like to say that during this relatively short time, the nanomedicine and nanotechnology in medicine already gained quite substantial or made substantial impact on the area. And in the majority of cases, it can be applicable for the development of novel or more advanced therapeutics or medical imaging concepts. Quite substantial improvement were developed in the area of biosensors, vaccine, and of course, it's a very interesting application in the regenerative medicine. But overall, what it's give us, so generally it will allow us right now and in the future to understand better the disease and most of all attack the disease and maybe even present it. But we're all interested in the oncology, in the cancers, and where the nanotechnology and nanomedicine can be applicable in this devastating disease. And first of all, generally we're talking about the drug delivery. So a lot of the pharmaceutical agents, they don't have a very good physicochemical properties, and that's why applying the nanoparticles as a vector for the delivery might improve the pharmacokinetic, might improve the more specific delivery to the certain organs, or sometimes even can be activated in terms of the activation of the release of the drug action using the different physical external stimuli. So of course we can use different targeting strategy, and here we can use the passive targeting, which is mostly till now was applicable in the oncology, using the pathological differences between normal and diseased tissue, or we can use the active targeting using some special receptors on the cancer cells to enhance the efficacy and specificity. So the other important application is how we can deal with the toxicity of the typical chemotherapeutics. And if we will apply the vectors for their delivery, we can either substantially lower the dose due to the specificity, or reduce the side effect through the, eliminating toxic solvents, or just kind of eliminating the timing of the infusions of the drugs, right? So, and if you look at on the material part of the side, so I kind of present on this slide, so how many different type of materials exist in terms of the useful platforms for drug delivery. So, and we're talking about the quite substantial difference in size, but what I would like to put your attention that if you look at the entities which are actively involved in the functioning of our biological system, they relatively represent the same scale in terms of size as a developed synthetic nanomaterials. And that is one of the driving force because this area could be just create the synthetic materials which will replicate or mimic the naturally existing forms. So, however, if you look at the nanomaterials, definitely they are more or less synthetic, and definitely they're much, much more complex compared to the drug molecule itself. So, first of all, we can use different type of the matter to create this type of entities. We can use the either inorganic or organic materials, we can use metal or lipids, but on the top of that, so we start to manipulate with the properties of materials at the level of the physical properties. Here we're talking about definitely size, shape, even the elasticity and, you know, kind of response to the stress or roughness will affect the biological behavior of that. So the other properties like surface of the materials, charge, functionality, or hydrophobicity, plus attachment of other molecules like targeting ligand. All of these properties will definitely affect biological behavior of such materials, their interaction with the proteins in the circulation, which will define the overall pharmacokinetic, biodistribution, tissue penetration. And generally, if you look even on this short list of the parameters, which we normally use for the characterization of the nanomaterials, you can understand that just to come to the final goal of improvement of the disease treatment, it's not easy task. That's why the translation of the nanomaterials into the medicine was quite challenging task. However, you know, if you look at that also from the point of how we can design it, we always need to think about major pharmaceutical characteristics. So biodegradability, so characterizations. So we need to look at the typical pharmaceutical properties like aqueous stability, colloidal dispersion, shelf life, and of course we're looking for the prolongation of circulation types to make it more available for the site of action, aggregation, many things. So despite of the complexity of this area, we still have quite substantial success in the area of application of nanocarrier into the cancer. So right now, already 16 different formulations are approved for the clinical application. So, and more than 75 of other ones are in the clinical trials. So if you look at the years, so the first formulation, nano formulation, was approved in the 1995, and the last one in the last year. So if you look at the diagram, of course, the most pronounced, most developed type of formulation, it's a liposome, it's different now. However, the other type of carriers like a micellar carrier or drug polymer conjugates also were explored and improved. So, and generally we believe that in the future, the major advantages of the nanomaterials in the oncology probably will lay in the area of combinational therapy, and we do have the first success in these areas. So recently it was approved liposomal double formulation for the application in the in leukemia, right? So or in the application in terms of multimodal therapy. And we're coming to the topic of the today event. It's how we can apply the nanotechnology to improve the radiotherapy. So, and there is a certain directions where it was explored. So first of all, to use this nanocarriers, nanoformulation for the radiosensitization. And starting for the approval of the first liposomal drugs, they were of course explored in the different clinical trials. However, no yet successful approval for the liposomal formulation in the combination with the radiotherapy is not coming from this trial. The other direction is definitely utilization of the nanocarriers in the chemoradiotherapy. And the third, which I will not touch, is generally how we can use the nano contrast agents for the better delineation if we're talking about the image-guided radiotherapy. So like I said, that quite a substantial number of clinical trials were performed. We don't have here yet the radiosensitizer in form of nanoformulation, which is approved in the combination with the therapy, radiotherapy. But one of direction is that could we apply this approach using the radiosynthesizers, which are not currently have a good pharmaceutical properties for their application. And one of the example which I decided to share with you is a molecule wortmannin, right? So this is what long time ago showed that it's very effective radiosensitizers. Why? Because it generally kind of inhibit the radiation-induced autophosphorylation of DNA-dependent kinases, which are kind of resulted in the radioresistance. However, these molecules have a very poor solubility, very low stability, and quite high toxicity. And it was obvious, could we just pack it in the nanocarriers? And this is just nanoparticles based on the PLGA. This is a biodegradable polymer, which is already approved by the FDA for the usage in the different devices. So it will covered with the lipid and pegylated for the better circulation. And that's how these particles are looked through the microscope. They're really tiny and they allow you, sustained release of the wortmannin from the carrier. Of course, the beta advantage, which was achieved, if you look at the MTD, you can see that there's substantial 5-time increase in the MTD of free drug compared to the nanoparticles. And preclinical studies were also very interesting. On certain type of the tumor xenograft, it was shown that definitely combination of the radiotherapy with a nanoformulated wortmannin provide much better suppression of the tumor growth. And what was also interesting, that it showed that molecular basis of the action also was proven, definitely by combination of radiotherapy with a nanoformulation, we see much less kind of resistance in terms of, or induced resistance in terms of the radiotherapy. What was also interesting, that this type of formulation behaved much better than typical chemoradiotherapy used in the treatment of the colon cancer. So in terms of survival, so the animals which were treated with the combination of 5-FU plus radiotherapy, so there were kind of less prominent survival compared to the group in which new type of the radiosensitizers and RT were used. So this might be one of the direction where we can go. However, the other interesting properties is, could we use the nanoparticles as a radiosensitizer themselves? And here we're coming to the metal nanoparticles, because it's quite known that materials, so named the Z-high materials, they can be kind of provide high absorption of X-ray, can be activated, and therefore, provide better dose enhancement, right? So the first material which was used was gold. However, till now, no clinical trials were successful. However, gold nanoparticles covered with the TGF-alpha right now in the clinical trials for the, kind of more like application in terms of induction of the chemotherapy, right? So, however, the other materials which was start to develop is hafnium oxide. These materials come to the under the microscope in 2003, and generally recently it was clinically approved. What it is? So hafnium oxides, it's also a Z-rich element. The particles of the oxide, they're relatively small, like 50 nanometers, covered with the polymer with the phosphate group, and generally have a negative size. And what is important, that these materials, they're inert themselves. However, they can be activated by radiotherapy. And as we knew before, so in the previous talk, generally there is a combination of the higher absorption, higher creation of the ROS, and overall it provides some improvement in terms of the response to the radiotherapy. What was also important, that it was shown that hafnium oxide nanoparticles kind of improve the response in terms of STING pathway, and that's why it can enhance the immunological response of the this treatment. So phase 2-3 trial was conducted in the Europe in the locally advanced soft tissue sarcoma. And what I'd like to say that in this case, the nanomaterials intratumorally injected. That's why it was shown that there is no leakage. So, and after that, the fractionated RT was applied before the surgery. Overall results with relatively high pathologic complete response, about 16%, and there was no additional toxicity compared to the arm in which only radiotherapy was used. And based on this result, the hafnium oxide nanoparticles were improved for the treatment of soft tissue sarcoma in Europe. And currently, the same approach are used in the multiple other clinical trials, particularly in the United States and the MD Anderson in a different type of cancer, as well as in the combination of radiotherapy with the PD checkpoint inhibitors in the patients with the advanced cancers. So this is a first nanomaterials, which is really clinically improved for the use with the radiotherapy. So the other type of the nanotherapeutics can also improve the chemoradiation. And here we'd like to present you the examples of the conjugate based on the competisin. So this type of molecules, they actually self-assemble in the particles. And since competisin is a very known inhibitor or radiosensitizers, and as well as inhibit hypoxia-induced factor, which is lead to the radio resistance. This type of conjugates went through the phase 1 clinical trial recently, and it was shown that generally its MTD can be around 15 milligrams, can deliver it weekly. Generally doesn't have any GI toxicity, and what is important that again, substantial number of patients reach complete response. So I believe that currently the phase 2 is planning for use this type of conjugate as in the combination of chemo and radiotherapy. As we know that radiotherapy can be also quite useful for the nanomedicine itself. You know that radiation is generally affect the tumor microenvironment in many ways in terms of alteration of vasculature, alteration of the extracellular matrix, and actually induction of the flow of the different lymphatic cells into the tumor. So, and what was shown that generally the initial treatment with the radiotherapy of the tumor potentiate the accumulation of the nanoparticles in the solid tumors. And most important, that recently mechanistically it was revealed that macrophages, tumor-associated macrophages, played quite substantial role in this process. They not only serve as a depot for the nanoparticles, they actually actively induce this tumor microenvironment response to the radiotherapy. And as you can see in this slide, so pre-treatment with the radiotherapy generally allow much deeper penetration of the materials into the tumor. And lastly, the other very interesting directions, which recently was described in the literature, could we use the nanoparticles in the radio immunology? In this particular case, it's a very elegant approach. What if we will introduce the nanoparticles inside of the tumor, then apply the radiotherapy, and generally these particles will capture the created antigen, near antigen, and then the nanoparticles can deliver it to the antigen presenting cells. And generally it was shown based again on the different type of the PLJ nanoparticles that yes, they can capture different number and different type of the proteins. And what is important that they do deliver it to the antigen presenting cells, and generally they significantly improve the efficacy of the checkpoint inhibitor treatment, and generally induce the good abscopal and direct immunologic effect. So, and my last slide, it's actually looking what might be a successful direction in terms of application of nanomedicine in the radiotherapy. No, there is definitely, we need to explore biology of the disease, right? How we can actually understand the interaction between the RT dose, between the time-dependent effect, and in context of very specific type of the nanomedicine. We have a huge problem and huge challenging in terms of how we can further develop a more rationally based construct. And last two questions, we still need to look at that. What will be regulatory consideration for applying this type of the treatment? And most of all, it's also important, what are the cost and what might be commercial potential? What will be the interest of the industry in terms of move forward this application? At this point, I would like to thank you and we'll be glad to answer questions if they will be.
All right, thank you, Bronich, for that very